Hello student, welcome to the new session of Anatomy of Flowering Plants. So this is the first session for this chapter, and in this chapter we will study about the anatomy, means the internal structure of the flowering plants. Okay, so let us start this session. So you can very easily see the structural similarities and variations in the external morphology of the larger living organism, whether it is a plant or an animal, right? So structural similarities and dissimilarities, that is also called as a variation. Okay, when you see the dissimilarities, that is showing the variation, right? So you can easily determine that this structure is very similar to in the flowering plants, and these structures are found to one particular type of plants or animals, right? So by just seeing the morphology, the external morphology, it is very easy to say that these are the similar structure that we see in the case of plants or animals, and these are the dissimilar or variation that is we see in the case of plants and animals on the basis of its morphological structure, right? So similarly, we were to study the internal structure, right? We have to study the internal structure. Internal structure is the study of internal structure of a plants and animals is called as anatomy. Okay, when you are studying the internal structure of plants and animal, then we can say that we are studying the anatomy of that particular plants. Okay, now one also finds several similarities as well as the differences. So as we have seen in the case of morphological characteristics, we do see the same thing in the case of anatomy of the flowering plants. So there are certain similar structure, right, in the case of either plants or animals on the basis of anatomy, similar internal structure, right? And also there are some differences. So you can make out that these particular structures are found to a certain type of plants only, and these are the certain structure that is present in almost in every plants, right? So on the basis of anatomy, you can find certain similarities and dissimilarities, or you can say the differences in the case of plants and animals, right? So in this chapter introduces you to the internal structure, right? So here we are studying the anatomy of flowering plant, then we are studying the internal structure and functional organization of the higher plants. So those internal structures are very important. They are doing some one particular function, right? So they are confined for the particular function, and also they are organizing the whole higher plants. Okay, so that we are going to study in today's session. So study of the internal structure of plant is called anatomy. If you are studying the internal structure of plants, okay, you cut the section of a stem, you cut the section of roots, you cut the section of a leaf, right, to see the internal structure, then you are studying the anatomy of that flowering plant. In the same way, we do for the animals also. We do for see the internal structure of animals as well, right? So we are studying the anatomy of the animals as well. Now plants have a cells as the basic unit. Okay, plants have a cell as a basic unit. Cell are organized into tissue, and in turn the tissue are organized into organ. So this is the basically the structural organization of plants. Okay, structural organization in plants. So we can see the different level. Cellular level means the plants are organized at a cellular level. Then there is a tissue level. We see the formation of different types of tissue. Okay, cells are aggregated together, or they are grouped together to form a particular type of tissue, and those tissue perform a particular function. So that is the formation of the tissue. Okay, and that shows the tissue level of organization. Now after that we see a different tissue Okay, different tissue organized to form organs. They are forming the organs as well. So we can see these structural organization in the plants, and the different organs in plants show the differences in their internal structure, right? So we can write some of the organs of the plants, right? We can see the leaf, we can see the stem, then we can see the root. Okay, and we see in all these three organs different types of tissue organization. Different organs plants show the differences in their internal structure. Means at the tissue level you see a lot of difference between these three organs. Okay, so the different tissue unite together to form organs. So these are the organs in the case of plants. Leaf is the organ, stem is the organ, root is the organ, right? And when you cut the cross section of the leaf, stem and root, you see a lot of differences at the level of organ. Okay, so different tissues are there to form these organs. Now within angiosperm, the monocot and the dicot are also seen to be anatomically different, right? So angiosperm in the last chapter, morphology of the flowering plant, you must have seen that angiosperm has been divided into monocot and dicot. So at the morphological level, you see a lot of differences in between the monocot and dicot, right? The cotyledons, on the basis of the cotyledons, you can divide this as a monocot and dicot. If the plant is having just one single cotyledon, then it is said to be the monocot plant, and the one that is having the two cotyledon, then it is said to be the dicot plant. On the basis of the root morphological structure, you can also differentiate the monocot and dicot. The monocot is having the fibrous root system, whereas the dicot is having the tap root system, right? And on the basis of the leaf venation, you can differentiate these two. Like in the case of monocot, the leaf is showing the parallel venation, and whereas in the dicot it shows the reticulate type of venation, right? And the flower, if you talk about the flower, then also you can see a lot of difference. The monocot, there are the trimerous, like 3 whorls you can see, whereas in the case of dicot it is tetramerous or the pentamerous, right? It means 4 or 5 whorls you can see. Now on the basis of the internal structure, when you see the internal structure of a dicot and monocot, then also you can see that this is a monocot plant and this is a dicot plant. It means that on the basis of internal anatomical structure, right, anatomical structure, you can differentiate the monocot and dicot. As you have differentiated the plants as a monocot and dicot on the basis of morphological difference, right? So in the same way, you can divide the plants as a monocot and dicot on the anatomical differences too, right? It means that monocot plant is a different anatomy than the dicot. Internal structure also show adaptation to the diverse environment. Why they are showing the internal structure so different? Okay, many plants show their internal structure is quite different from the other, and this is just because the adaptation, because they have to live in a particular environment. Okay, there are so many diverse environment, so they show the adaptation in those diverse environment, right? So we were discussing the type of tissue based on the type of cell present. Okay, there are different types of tissue, and these type of tissue, they are present on the type of the cell present in their tissue, right? So let us see that. So let us now consider how tissue vary depending on their location in the plant body. So this is the tissue system. We are studying the tissue system, right? tissue system. So let us now consider how the tissue vary depending on their location in the plant body. Tissue is varying, okay, they are varying their types. So their structure and function would also be dependent on the location. So the tissue, they are having a different location in the plant body. Different types of tissue is having the different location according to the organs they are having, and also the structure and function, right? So on the location, you can have the different structure, and it is related to the function, like the function of root, the specific type of tissue that govern for its structure and its function is present in the root only. It will not be present in the stem. And in the same way, the structure, the tissue that is present in the stem, right, it will be quite different from the one that is present in the root system, because the root is having the different function, it will have the different structure. The stem is having the different structure, so it will have the different function, right? So on the basis of their structure and location, there are three different types of tissue system. Okay, so we are having the three different types of tissue system. So there are the epidermal tissue system, right, the ground or the fundamental tissue system, and the vascular or the conducting system, right? So there are the three different types of tissue system. The number one is, that is the epidermal tissue system. Okay, the second one is the ground or the fundamental tissue system, and the third one is the vascular or the conducting tissue system, right? Now if you talk about the epidermal tissue system, the epidermal tissue system forms the outermost covering of the whole plant body and comprises the epidermal cells. Okay, so this particular tissue is present on the whole body, whether it is a stem, whether it is a root, whether it is a stem. You can see this epidermal tissue is present on the all the surfaces of the organ, right, of the plant, right? So and that is making the outermost covering of the whole plant body. Okay, this is making the outermost covering of the whole plant body, and it also comprises the epidermal cells, stomata, and the epidermal appendages, that is the trichomes and hair. So you can see these structures. So let us see these structure on the sorry, blackboard structure. So here we can see the epidermis. Okay, single layer. It is, if it is present on the leaf, then you can see the epidermis is present on the upper surface and lower surface as well. So it is making the upper epidermis and the lower epidermis. So suppose this is, I'm drawing the cross section of a leaf, then this is the, okay, lower surface. Okay, this is the lower surface. So these are what? These are the stomata. You can see the stomata structure is here. Okay, these are the stomata. So this is the modification of the epidermis. What are the stomata? Stomata are the openings. These are the openings. And this is meant for the exchange of gases in plant, right? It is meant for exchange of gases. Exchange of gases in plants. And again, this is a modification. This is the modification of epidermis, epidermal cells. In fact, this is the modification of epidermal cells, right? So these are the stomata, that is the openings. And at some places you can see the hair like a structure. Again, this is, I'm drawing the epidermis. Okay, so you can see some hair like a structure like this. Okay, these are what? These are trichomes. Trichomes are multicellular structure. Sometimes they are just hair like a structure, and sometimes they are glandular also, secretory in function. They are sometimes secretory in functions as well, right? So these are what? These are the modification of the epidermal tissue, right? So here what we have seen here, the last point, that the epidermal tissue forms the outermost covering of the whole plant body, and it comprises the epidermal cells, stomata and epidermal appendages. So you can see the stomata is a modification of the epidermal cells, and some epidermal appendages also, the trichomes and hairs. The epidermis is the outermost layer of the plant body, right? So this is making the outermost layer of the plant body, and it is made up of elongated, compactly arranged cells which form a continuous layer. So this is making the continuous layer, and there is no space in between the cells. If you see the epidermal cells, you will not find any type of cells in between. Okay, the cells are quite elongated structure and they are compactly arranged. This is the characteristic of the epidermal cells, right? And they are continuous layer. You won't find any a gap type of things, right? You can find the modification but not the gap. So it is making the continuous layer, and epidermis is usually single layered, right? This is also one of the very important point, that epidermis is making the single layer. So you can see the single layer of epidermis on the surface of the leaf, on the surface of the stem, and also on the surface of the root, right? So epidermal cells are parenchymatous. If we talk about the what type of cells that make the epidermal tissue, then we can see that it is made up of the parenchymatous cell with a small amount of cytoplasm lining the cell wall and the large vacuole, right? So you can see the small amount of cytoplasm is very less, and it is having the cell wall and a large vacuole. So if I draw one of the structure of the epidermal cells, right, it is somewhat like this. So this is the cell wall, this is the cell wall, and this is the plasma membrane. Right, now the vacuole is large, nucleus is here, right? And if you talk about the cytoplasm, then cytoplasm quantity is very
very less. The whole cell cytoplasm is very less in amount. Major part of it is covered with a large vacuole. Okay, a large vacuole is found. So this is very important. And the outside of the epidermis is often covered with a waxy thick layer of layer that is called as the cuticle, which prevent the loss of water. So at the surface, what you see, there is a deposition of a waxy layer at this, and that is making the cuticle. Okay, so this is the cuticle at the surface of the epidermis. So this thick or waxy thick layer, the cuticle, it prevents the loss of water. It doesn't allow the water to evaporate from the cell. Okay, and cuticle absent in the root. If you see the epidermal cells, okay, the epidermal cells of the root, then you will see there is no cuticle found on its surface, because the water loss is occurring on the from the roots, from the leaf structure, from the stem structure. So this cuticle is present at that surface, not on the root, right? So this you have to remember. Stomata are the structure present in the epidermis of the leaves. No, this is the structure that is present on the surface, in which surface? Epidermis of the leaves. And the stomata regulate the process of transpiration and exchange of gases. Whatever the excess of water, right, is there in the plant, that get evaporated, and that evaporation of water from the surface of the leaf, okay, when the stomata get open for the gaseous exchange, right, then excess of water get evaporated, and that is called as transpiration. Okay, so when there is a lot of water, so stomata get open to let the water goes off, some of the water, okay, and it also causes the gaseous exchange, right? For the process of photosynthesis, there is a requirement of the raw material in the form of gases, that is a carbon dioxide. So carbon dioxide is taken when this stomata opens, right? And at the same time, the previous photosynthesis which has been occurred earlier, and due to that a byproduct in the form of gases is formed, that is oxygen gas, okay, so that has to be removed out from the cell. So carbon dioxide is taken inside and the oxygen is given out. So this is the gaseous exchange that is taking place through the stomata structure, right? So it is regulating both this transpiration and the gaseous exchange. Now each stomata is composed of a two bean-shaped cells known as guard cell, which encloses the stomatal pore. So if you see the structure, okay, if you see the structure of a stoma, then you can see it is made up of the bean-shaped cells. The shape of the cells is quite a bean shape, and that is called as a guard cell, and it is enclosing the stomatal pore. So in grasses, the guard cells are the dumbbell shapes. Okay, we will see the structure. We will see the structure of this tissue in just now in a slide. Okay, so before that, let us see what type of stomata cell is found in the grasses. That is, the grasses belongs to monocot. It belongs to monocot. So the guard cells are dumbbell shaped. So if you see the guard cells, it is somewhat like this, dumbbell shape, right? Dumbbell shaped guard cells we see in the case of, this is the guard cells, okay, and this is the opening. So this type of stomata is found in the case of the grasses. Now the outer wall of the guard cells away from the stomatal pole are very, very thin. Outer wall of the guard cell. So this is the guard cell in the case of dicot. Okay. It is very thin. This is thin. And what about the inner one? This is quite thick. Okay. So this is the thick. This is both. Okay. Toward the, it is the thin, and the inner walls are highly thickened. So this is the outer wall of stomata, outer wall of the stomata, and this is the inner wall. This is the inner wall of the stomata. Inner wall of the stomata, right? So the guard cell possesses the chloroplast. You can see the guard cell is having chloroplast in it, and it regulates the opening and closing of the stomata, right? It is regulating the opening and closing of the stomata. So the guard cells are very, very important, as it possesses the chloroplast. It means that in the case of guard cells, the photosynthesis is taking place. Okay, the sugar is synthesized, and it regulates the opening and closing of the stomata as well. So this is very, very important, as guard cell. Now, sometime a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known as subsidiary cells. So
few epidermal cells that are very near it, in the vicinity of the guard cell, means the cells which are very close to the guard cells become specialized. It becomes specialized in their shape and size and are called as subsidiary cells. They are called as subsidiary cells. So here you can see the both the type of stomata. Okay, the stomatal aperture, guard cells, and the surrounding subsidiary cells are together called the stomatal apparatus. So guard cells, subsidiary cells, okay, these two together it is called as the stomatal apparatus. So what you see here, this is the outer surface of the guard cells, right, and it is quite thin. The inner one, that is, you can see the very dark, that is quite thicker one, and you can see the chloroplast here. The chloroplast, these are the chloroplast that is present in the guard cells, so it means that it is performing the process of photosynthesis. And here you can see the dumbbell shaped guard cell. This is the dumbbell shaped guard cells. Okay, dumbbell shaped guard cells 1 and 2, and stomata with the dumbbell shaped guard cell. This you see in the case of grasses. And you can see the subsidiary cells, the surrounding cell 1, 2, 3, 4. So 4 cells surrounding these 2 guard cells, and it is unitedly that is called as the stomatal apparatus. Means subsidiary cells and guard cell unitedly it is called as the stomatal apparatus. So here also you can see in the case of this 2 guard cells, there are the 2 subsidiary cells, okay, both the side, and this we see in the case of grasses. Now the cells of the epidermis bear a number of hairs. They are also having the numbers of hairs on their surface, and the root hairs are the unicellular elongation of the epidermal cells and absorb water and mineral from the cell. Now if you see the structure of the root hairs, right, so the epidermal cells give rise to the hair like a structure like this. Okay, so this is the root hair. This is the root hair. So this is again the epidermal cells. Epidermal cells in the case of root, epidermal cells. Okay, so this root hair that helps to increase the surface of water, surface area, and it increases the absorption of water and minerals from the soil. And one of the stem, the epidermal hair, that is called as trichomes, and we can see some hair like a structure that is coming out from the surface of the stem, that is also called as the trichomes. Now these trichomes in the shoot systems are usually multicellular, whereas in the case of root system, you see the epidermal cell give rise a unicellular root hairs, right, whereas in the case of the shoot system, these trichomes are multicellular. They may be branched or unbranched and soft or stiff. So sometimes these cells are branched and sometimes these cells are unbranched. They are soft or stiff. They may even be secretory. Sometimes these trichomes are secretory in structure, right? These are secretory in structure. The trichomes help in preventing water loss due to transpiration. So these trichomes are helping in the prevention of water loss. They do not allow the extra water to get lost from the surface of the stem. Okay, so this trichomes will helping the preventing of water loss due to the transpiration. Okay, so this is all about the epidermal tissue. Now we are having a ground tissue system. So the all tissue except the epidermis and the vascular bundle constitute the ground tissue, right? So just leaving out the epidermis and the vascular tissue, whatever the tissue is present in between that, it is called as the ground tissue. So let us see the ground tissue. Now it consists of the simple tissue such as the parenchyma, collenchyma and sclerenchyma.
right? So if you talk about the parenchyma cell, then parenchyma cells are isodiametric like this, and they are having their intercellular spaces, they are having their vacuoles. Okay, so we can see different types of parenchyma, and there are some certain modifications as well. So here you can see the intercellular spaces, okay, they are basically meant for the storage, but sometimes they do contain the chloroplast, so they do the process of photosynthesis also, and at that time these parenchyma cells are called as chlorenchyma. Okay, now sometimes they are having the large air spaces in between that. Okay, and these large air spaces form a new type of tissue that is called as arenchyma. Okay, parenchyma. Now, parenchymatous cells are usually present in the cortex, pericycle, pith, and medullary rays in the primary stem or root. So if we see the primary structure of the root and stem, we can see this parenchymatous, the first type of cells. The parenchymatous cells, they are present in the cortical region, they are present in the pericycle region, they are present in the pith as well as in the medullary rays. In leaves, the ground tissue consists of thin-walled chloroplast containing cells, and it is called as a mesophyll cells, right? So if you see the ground tissue, then it is the cell, the wall of the cells are very thin. It is a chloroplast containing, and it is in the case of the forming the mesophyll cells. So this is in the case of leaves. So these places you can find this parenchymatous cells. Vascular tissue system. If you see the vascular tissue system, then the vascular system consists of the complex type of tissue, very complex type of tissue, phloem and xylem, and the xylem and phloem together constitute the vascular bundle. So let us see what are these complex type of tissues. In case of plants, so we are having two different types of tissue, xylem and phloem. Now if we see the xylem, what are the component of the xylem? The component of the xylem are xylem vessels, xylem tracheids, xylem parenchyma, and the last one, that is the xylem fibers, right? So these are the four components of the xylem, and out of this, the component that is the xylem vessels, xylem tracheids, and xylem fiber, these are dead, they are not living. Whereas this one, the xylem parenchyma, this is the only living cell that is present in this complex tissue, that is the xylem. All are dead, right? And they are very important for the absorption of water from the roots, okay, and also transporting it to the upper parts of the plant. Not only the water, but they are also absorbing the minerals along with the water. Now, if you see the component of the phloem, then phloem is having tube cells, subsidiary cells. No, this is the companion cells. Then they are having phloem, fibers, and phloem
parenchyma. Phloem parenchyma. So if you see this tube cells, companion cells, and the phloem parenchyma, okay, these three, tube cells, companion cells, and phloem parenchyma, these are the three living, just opposite. There the three were dead, only one living. Here three are living and one dead. That is, the phloem fiber is dead, whereas the three are living. So this is the component. Earlier the tube cells were, when it is present at the time of formation, okay, at the time of origin, it is having the nucleus. But later what happened, this the nucleus of this tube cells, it degenerates, and whatever the component is required for like from the nucleus, it is given by the companion cells, right? So this companion cells and tube cells, they are originated together, and later they form two different cells, but they are helping each other in that respect. So that is why this cells got the name companion cells. So here you can see the vascular tissue. Okay, so vascular tissue, here you can see it is formed in the form of the vascular bundle. This you can see in the form of vascular bundles. Now with the vascular bundle, you can see those two components, the xylem and phloem. Here the xylem is present on the one radii, and it is present like this. Okay, it is present at the one radii, the small at the gaps, and that is showing the xylem. Now phloem, you can see it is present towards the periphery at another radii, right? So this is showing the radial type of arrangement of the vascular bundle. Okay, radial type of vascular bundle arrangement. Now on the basis of the present of this is the conjoint. Okay, these xylem and phloem at present at the different radii, Now here, when they are present on the same radii, they are compact together. It is called as conjoint type. Conjoint. Okay, you can see the xylem here, and it is upper portion that is showing the phloem. So they are not present at the different radii. In fact, they are very close together and at the same radii. And you can see in the case of this B structure, there is no in between cells, right, between this phloem and xylem. So no vascular cambium tissue is there. No cambium tissue is there. Okay, no cambium. Here you can see no cambium. Whereas in this form, C, that is the conjoint open, you can see the cambium is present. So when the cambium is absent, the conjoint vascular bundle is called as closed type. Okay, it is called as the closed type of vascular bundle. Whereas in the case of vascular bundle where the cambium is present, it is called as the conjoint open. The vascular bundle is open type. So in dicotyledonous system, the cambium is present between the phloem and xylem. Okay, when you see the dicotyledonous system, you can see the cambium is present between the phloem and xylem, and such vascular bundles of the presence of the cambium possess the ability to form the secondary xylem and the phloem tissue, and hence called as the open vascular bundle, right? So this vascular bundle, this is having the cambium, so it is called as the open type of vascular bundle, and this cambium is having the capacity to divide and redivide and form a new type of cells. So it can form secondary xylem and secondary phloem, right? Secondary xylem and secondary phloem. And this cambium is a type of meristematic tissue. Meristematic tissue. You must have studied in the class 9th the different types of meristematic tissue, that is the apical meristem, okay, intercalary meristem, and the lateral meristem. So this cambium is the lateral meristem. This is just the lateral meristem, and it divides and redivides the cells and forms the secondary xylem and the secondary phloem.
Now, in the case of monocot, the vascular bundles are not having any cambium in them. No cambium is found. So that is the closed type of vascular bundle. Just now we have seen in the previous slide, closed type of vascular bundle, and it is not showing any type of secondary growth, right? Hence, since they do not form the secondary tissue, they are referred to be closed. And in the one that in the case of dicot, the vascular bundles are open, whereas in the case of monocot, the vascular bundles are closed type. When the xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called as the radial, such as in the case of roots. So if we draw the structure of a radial vascular bundle, so you can see the xylem. The xylem is present at the one radii. Okay, it is like this. This is the xylem. And the phloem part you can see here. This is the phloem part. So this is the phloem, and this is the xylem. This is the xylem, right? So the conjoined type of vascular bundle, the xylem and phloems are joined, situated along the same radii of the vascular bundle, just opposite to this one. In the case of conjoined type of vascular bundle, you must have seen in the previous slide, right? It is like this. This is the xylem, and you can see the phloem here. They are present at the same radii, right? So here you can see the phloem is at other radii, the xylem is at other radii, but here the xylem and phloem, this is the phloem and this is the xylem. They are present at the same radii, so this is called as the conjoint type of vascular bundle, and this is the radial type of vascular bundle. Such vascular bundles become common in the stems and leaves. So conjoined type of vascular bundle is very common in the case of a stem and the leaves. Now the conjoined vascular bundles usually have the phloem located on the outer side of the xylem. Okay, this is a very important point, where the phloems are present. So phloems are present on the outer side of the xylem. It is present on the outer side of the xylem. Okay, in the case of conjoined vascular bundle. And for a better understanding of the tissue organization of root, stem and leaves, it is convenient to study the transverse section of the mature zone of these organs. Okay, so to see the tissue organization of the root, stem and leaves, it is very convenient to study the transverse section. So if you want to study the internal structure of stem, so you will see the cut section of the stem. If you want to see the internal structure of root, then you will see the cut section of the transverse section. It means the transverse section of the root, and the same way in the case of leaves also. So let us see here dicotyledonous root. So look at the figure 6.3. It shows the transverse section of the sunflower. So here you can see the structure. Okay. So here you can see the xylem, this red color point. This is the whole vascular bundle, right? In the vascular bundle, what you see here, this is the xylem. Okay, these are the arrangement of the xylem, and here you can see the phloem. Okay, here you can see the phloem like this. Okay, they are present at the different radii. Okay, and you can see the cortex region that is made up of the
parenchymatous cells, intercellular spaces, and here you can see the root hair. The epidermis is giving right there the root hairs, right? Now this is the monocot. This is the dicot primary root, and here you can see the monocot. Okay, they are present at the different radii. So you are having this phloem region, okay, phloem, and you can see the xylem, protoxylem and the metaxylem here, right? And in the center you can see the pith, okay, and the cortex region. You can see the cortex region. Again, it is made up of the same type of parenchymatous cells, and the root hairs, this is coming out from the outer epidermis. Now the internal tissue organization is as follows. The outermost layer is epiblema. Okay, this is epidermis. When it is present on the surface of the root, it is forming the epiblema. That is called as the epiblema. And many of the cells of the epiblema protrude in the form of unicellular root hairs. So epiblema is giving right the root hairs. And the cortex consists of the several layers of the thin wall parenchyma cells and the intercellular spaces. Okay, so the cortex is made up of many layers. You can see the different layers of this parenchymatous cells. That makes the cortex region. The innermost layer of the cortex is called as the endodermis. So very important. The outer epidermis, just below the epidermis is start the cortex layer. And till where the cortex layer is? Till the endodermis layer. Okay, now it comprises the single layer of the barrel shape without any intercellular spaces in between. So no intercellular spaces you can see in the case of the endodermis. The tangential as well as the radial wall of the endodermal cells have a deposition of the water impermeable wax material suberin in the form of the Casparian strips. This is again very important. So this endodermis is having this deposition of the waxy substance that is called as suberin, that is making the Casparian strips. Okay. Next to the endodermis lies the few layer of thick wall parenchymatous cell that is referred as a pericycle. Now this pericycle is not the part of the cortex. Okay, cortex is still the endodermis only. Now the initiation of the lateral roots and the vascular cambium during the secondary growth take place in this cell. So pericycle is very important, right? Lateral roots, many secondary roots comes out. Root hairs are coming from the epiblema cells. That is the modification of the epidermis, right? But the lateral roots, it's coming from the pericycle region. It is coming out from the pericycle region, and it is making the vascular cambium during the secondary growth that take place in the cells. The pith is a small or inconspicuous. The pith is very small in size. Okay. The parenchymatous cells which lie between the xylem and phloems are called as the conjunctive tissue. Okay, so this parenchymatous cell which is present between the xylem and phloem, it is making the conjunctive tissue. Very important. And there are usually 2 to 4 xylem and phloem patches. In the case of the vascular bundle, when you see in the center, so you can see some patches of the xylem and phloem. So how many patches? 2 to 4. Later, the cambium ring develops between the xylem and phloem. So what happened? You can see the vascular bundles are like this. This is the endodermis. Vascular bundles are like this. At the time of secondary growth, what happened? The cells in between the vascular bundle, they get initiated and transformed it in a form of cambium, and it lately making the whole cambium ring like this. Okay, so normally the cells in between this, they are parenchymatous, but at the time of secondary growth, these cells between, that is the phloem and xylem, the conjunctive tissue, okay, so they get transformed and making the cambium ring. So here you can see that earlier the cambium is just present in between the vascular bundle, okay, in the vascular bundle. So here you can see the cambium, but later in between the vascular bundle, this layer, this is forming the intervascular cambium, and it is making a totally a ring like a structure like this. So that is making the cambium ring that develops between the xylem and the phloem. Now all tissue on the inner side of the endodermis such as pericycle, vascular bundles and pith constitute the stele. Okay, this is called as the stele. So from the pericycle, okay, till the endodermis, it is the that is the ground tissue, that is the cortex region, okay, and from the pericycle, okay, including the vascular bundle and pith, making the stele region. So this is about the dicotyledonous. Okay, let us see about the monocotyledonous root now. The anatomy of the monocot root is similar to the dicot root in many respects. So if you see the figure, okay, so monocot root you can see here, it is very much similar. It is not showing much difference between the dicot, right? So it has epidermis, it has a cortex, endodermis, pericycle and vascular bundle and pith. As compared to the dicot root with a favored xylem bundles, the xylem bundles are less in number and they are usually more than 6, that is the polyarch xylem bundles in the case of monocot. Okay, so dicot, they are showing the favored number of xylem bundle, whereas in the case of monocot you can see the bundles are quite large in number. Okay, that is the polyarch, that is possibly be the 6 in number, polyarch. Now pith is large as and well developed in the case of monocot, and monocotyledonous root do not undergo any type of secondary growth. No secondary growth is seen in the case of the monocotyledonous root, right? So this is all about the anatomy of the dicotyledonous and the monocotyledonous plant. So this is the end of this particular session. We will have one more session to complete this whole topic. Till then, thank you very much.